Hi guys, in this video we're going to talk about the histology of the GIT. Here we will start with the esophagus and stomach, and then we're going to talk about the small and large intestine in a separate video, and then the glands and extramural parts of the tract, the organs that are going to help in digestion, which are out of the tract itself. It's going to be in a separate video. Now, to talk about the alimentary canal, we need to understand the basic components of that canal. So basically, if you guys look at the canal itself, you will find that it's separated into four parts. The first part is actually the mucosa itself, and then it's followed by the submucosa, and then you have the muscularis, which is inner circular and outer longitudinal, and then outside you have the serosa, which is a connective tissue. And if you are in a part that is not covered by the visceral peritoneum, you don't have that layer of mesothelium, we call it adventitia. Now, every one of them has its own importance, and because structure explains function, if we look at the different parts of the canal itself, or the tract, of the GIT tract, and try to understand why the structure looks like that and try to relate it to the function, it will be very easy to understand it and identify it later on and study it. Now, if you look at the mucosa, so now we said that we have mucosa, we have submucosa, we have muscularis, and then serosa. 1, 2, 3, 4. Now, the mucosa itself is further divided into your lining epithelium, which is followed by lamina propria, and then muscularis mucosa. Why do I have an epithelial lining? Why do I need to have cells? Because this is my GIT. I'm eating food. I want to digest the food. I need to absorb the nutrients out of the food. I want to secrete mucus, for example. I need to secrete agents that will help me in digestion. So I have functions, and this is the very first layer that's gonna face my lumen, and it's gonna face my food, and it needs to adjust itself in such a way it helps me to carry the function. And then that is the epithelial lining, and based on the function, we're going to see how the cells of the epithelium of different parts of the GIT are going to look like. Now, that is followed by the lamina propria, and that lamina propria is actually your connective tissue that follows your epithelium. So if you need to have lymphoid aggregations, if you need to have lymph cells, if you need to have immune cells, if you need to have innervation that reaches there, blood supply. So everything is going to be ensured by the lamina propria, and thanks to the lamina propria connective tissue that supports the epithelium. Because by definition, the epithelium is avascular. It lacks a lot of important stuff, and that is going to be ensured by the lamina propria. So it's basically the connective tissue that supports the epithelium itself. All right, and this is followed by the last layer, which is muscularis mucosa, and that is basically a muscular layer. And this muscular layer is actually very important, because as this muscular layer contracts, you think about this canal, and then you have here a muscular layer below your epithelium. If this muscle layer contracts, it's kind of creates kind of crypts or valleys or depressions and ridges. And when that happens, think about it, it helps you to absorb, yeah, because this is your first epithelial layer here. This is a columnar cell, let's say, and now you need to kind of contract and kind of create these ridges down here, very, very following the epithelial lining, and that is going to help you to absorb and secrete also as well, because it gives you the opportunity to be kind of exposed to the lumen. All right, so this is why it's important to have the muscularis mucosa, and it also helps to if there is secretive or secretive cells, it helps to kind of empty the secretion of these cells into the lumen. Now, followed by submucosa. Submucosa, it's important because this is connective tissue, and you guys know that as basic components of the histology, we have four basic types of tissue. We have the epithelial tissue, we have connective tissue, we have muscle tissue, and nervous tissue. Now, as I'm moving from one type to another type, I need to have these connective tissue that connects all the basic tissues together to give me the organ. So if I have epithelium and I want to move on to another type of tissue, which is a muscular layer, I need to have a layer of connective tissue. And that is here in the GIT, we call it submucosa. So submucosa is nothing but the connective tissue that is sub the mucosa, which is below the mucosa, that's going to support it, that's going to have, again, important structures that will help you, for example, to have the vasculature, the innervation, the lymphatic nodules, the lymphocytes, the plasma cells, whatever you need for your mucosa. And also it will help you to connect the mucosa to the next muscularis layer. All right, in the submucosa sometimes it has glands. All right, and that is the alimentary canal. When we talk about the alimentary canal, we have submucosal glands only in two regions, and these regions are the esophagus and the duodenum. All right, so remember that the submucosal glands are only there in the esophagus and the duodenum. Other than that, you don't get to see it, but you get to see lymphatic nodules, and lymphatic nodules is extensively there. Like if we talk about the mucosa, the glands, the submucosal glands. All right, we get to see them in these places. And if we talk about lymphatic nodules, they are dispersely there everywhere, but you see them extensively in the ileum, all right, and the appendix, all right, and also it's there in the colon, in the large intestine. And that we get to see it where? In the submucosa. All right, this is a general introduction to the histology of the GIT. Now, this is followed by the muscularis externa. So in the muscularis externa, why do we say it externa? Because we have muscularis interna in the mucosa itself, and we talked about it. Now, why do we have muscularis externa? It's actually the inner circular and outer longitudinal layers. Why do we need to have two layers? Now, if we think about the muscularis externa, it's basically, generally, it's the inner circular and outer longitudinal, all right, longitudinal layers. And in the stomach, we have also oblique muscles, because in the stomach, it's very busy organ and we really need to mix the food very well. So we need to have a lot of directions of the muscle fibers that will help me to kind of squeeze that organ and get the food mixed and do the great job that's happening in the stomach, actually. Now, why do I need to have inner circular? Because I need to mix the food in the GIT. Now, why do I need to have outer longitudinal? Because I need to propel the food from one place to the next, from one place to the next, from one place to the next. All right. So thanks to the inner circular, that by the contraction of the inner circular, I will make sure to mix the food very well inside my canal. And thanks to the outer longitudinal, that will help me to proceed that time from one place to the next, to get the food processed from one area to another and get it done all the way, digestion and absorption, getting the nutrition from the food I'm eating, and getting the benefit of why I'm eating that food. All right. In some places, we have modifications of one of these two layers of muscularis externa. I want you guys to relate this histological information to the gross anatomy. So in some places, we have modifications on these, one of these layers becomes developed in such a way, it makes another function. And that is an example for that is in the inner circular, it's the same layer that gets modified and forms a sphincter. So the pyloric sphincter is actually coming from this layer, modified, formed in a fashion that causes the pyloric sphincter that will make sure to guide the food as it comes out from the stomach to the duodenum. Okay, on when exactly we should keep it closed as we are digesting the food, and where exactly we should proceed and push it to the duodenum to continue digestion of the fat, for example, and the stuff that is going to be taken care by the duodenum itself. For the outer longitudinal, we have also a modification as these muscles send fibers to form the tinea coli. So the tinea coli that are wrapping around, if you guys look at this, can you guys, let me just try to delete here. Can you guys see this? Can you guys see this? Okay, so this is actually the tinea coli. This is actually a modification of the outer longitudinal layer that is going to be there in the colon, and by contraction of this, that will help also to propel the food, and that will help in digestion and movement of the chyme from one place to another, shortening the colon to fit into the abdomen and so on. So these are basically gross anatomical features that are actually related to some histological features in the GIT system. We're going to move on and talk about the histology into details now, starting with the esophagus. But I'm going to stop the video here and I'm going to do another video because now you have a general idea about what we're going to see. And now we want to start another video and talk about the histology of the esophagus and proceed to the next part. So see you guys in the next video.